All right, everyone, here we are proceeding to our new adventure of, well, not new adventure, but continuing our adventure in our discussion of carbohydrates. So, in our first exploration of carbohydrates, we were really kind of focusing on individual sugars. And, we learned about how they can be modified, types of linkages they can form. And in this one, we're going to dive deeper into that. We're going to talk more about, well, how those linkages can produce more complex carbohydrates. So let's go ahead and get started. So the first class, and right off the bat, of carbohydrates that are more complicated than the sugars we were talking about are a class of compounds known as disaccharides. And one example of a disaccharide is a molecule known as lactose. So when you look at any sort of sugar or disaccharide, it's important to be able to look at it and basically say what it is. So, what monosaccharides are contained in lactose? And these are all questions that I want you to be able to answer for your exam. What monosaccharides are contained in lactose? What isomeric forms of each monosaccharide is present? How are these monosaccharides linked together? What positions of each monosaccharide are linked? What is the name of the disaccharide? And what is the name of the enzyme that cleaves lactose? Okay, so if you remember, there were a total of six aldoses that I wanted you to know the structures of. And, I talked about if I were to triage them, the highest on that would be glucose. And so here's my linear form of glucose. I'm going to make first my 5, 6 carbon linear chain of carbons. So here's my linear chain of 6 carbons. I'm going to put an aldehyde functional group on carbon number 1. And at this point in time of the structure that I've drawn, none of these carbons, well, are chiral. However, whenever I draw glucose, and the pattern that I always think of whenever I'm drawing glucose is I go to my carbon, which is farthest, or my chiral carbon, which is farthest away from my functional group, and that would be this one. I've got my hydroxide group on the right-hand side, indicating that I'm drawing a D form of glucose. And for me, glucose, the way that I remember it is right, left, right. I'm putting a hydroxide group on the right-hand side of carbon number 2, left-hand side of carbon number 3, right hand of carbon number 4. So I've got H, H, H. So I've got right, left, right, and then hydroxide group on the right-hand side because it's a D sugar. Now, with that in mind, whenever I translate this from this linear configuration to the cyclic configuration, my right, left, right becomes down, up, down. And I'm going to number my carbons off here, and then I'm going to draw the ringed form of glucose. So 1, 2, 3, 4, 5, and 6. So there is, or there are my 6 carbons of my glucose ring. Now, carbon number 1 is right here. And I gotta draw it with my hydroxide group pointing downward, which whenever I do that, I've made an anomeric variation or an anomeric form of glucose. I drew that down, so this is the alpha form. Now, I'll kind of be candid for y'all. Whenever you're drawing a D sugar, or whenever you're drawing a ring sugar, the L variations of your sugars are fairly uncommon. So we're looking at the D version of this sugar. The convention would be to draw that D sugar because that's the biologically relevant form. Now, your L sugar, can it exist? Yeah, absolutely. Can an L ring version of a sugar exist? 100%. Now, how would you draw that? Basically, what you would do is you would do exactly what you do when you're drawing the mirror image of your L sugar. And that is you're going to take every single one of these points and basically flip them. In addition to that, you're going to draw that ring basically backwards. So rather than this being outside of the ring, instead it'd be down here. So for for our purposes, I always like to kind of go from start as broad as possible, but then narrow it down what we're talking about to the more biologically relevant information. So we are drawing a alpha D glucose. Okay, so the next thing that I want to talk about is how do I take, so I've accounted for carbon number 6, 5, okay. 1 is now a chiral center, so I've accounted for 1, 5, and 6. All of those have groups bound to them. Carbons 2, 3, and 4, however, do not. Now, what I'm going to do is I'm going to translate right, left, right to down, up, down. So right or down, left, up, down, right. So OH, OH, OH. So there's my structure. There is the form of glucose that I wanted to draw. Now, let's look at that and let's compare it to my rings up here. So I've got ring form A. Okay. Now, what is the difference, or sorry, monosaccharide A and monosaccharide B? So A, what's the difference on carbon number 1 and carbon number 1 of my ringed glucose? Well, 1, I've got this funky bond that's going to an oxygen, which, spoiler alert, that is there. This is not a carbon and this is not a carbon. So this is just carbon number 1 goes to oxygen, much like this does. Okay. The difference is that carbon number 1 on the sugar that I drew, it's pointing downward, whereas B, it's pointing, or sorry, the disaccharide, it's pointing upward. So it's a different anomer. Okay, so that's not really too much of a difference. The second item would be carbon number 2. Well, that's also pointing down on my ring as it is in in the disaccharide, but also in the ring that I drew. What about carbon number 3? That's pointing upward, so those are the same. Carbon number 4, however, there we have some descent. Carbon number 4 is going to be different. So this is what would be known as the C4 epimer of glucose. Now, if you were to go back and look at the sugars that I wanted you to know the structures of, which I'm going to switch my share real quick. Come on, computer. There we go. The C4 epimer of glucose. Okay, so glucose, I've got that. And then the C4 epimer. Well, that is going to be galactose. Because if you look at the structures of glucose and galactose, glucose here on the, I've lost my annotate, there it is. So glucose right here, galactose right here, C2 right here, those are identical. C3, those are identical. But C4, that's where we have descent. That's where we have a difference between our sugars. So now I'm going to switch my share back. And I'm going to pause real quick. So this sugar on the left is galactose. Now on the right, let's look at that. Okay, so on the right-hand side, my B sugar. Well, carbon number 1, that's the same. Carbon number 2, that's the same. Carbon 3, the same. Carbon 4, okay, this is going down, so that's the same. So lactose, so question number 1, what monosaccharides are contained in lactose? Galactose and glucose. Okay. Okay, so what isomers? Well, we've got a beta configuration and an alpha configuration of our sugars. They're both D sugars, so we're good there. Now, how are these monosaccharides linked together? Oh, this is a great question because and I'm going to clear most of my drawings. We have maybe one of the most important parts about sugars, the linkage. This is a glycosidic linkage, but more specifically, the way that we describe it is we say that this is a beta 1, 4 glycosidic linkage. So beta 1,4. What positions of each monosaccharide are linked? Well, that goes back to our previous one. This is a linkage between carbons number 1 and carbon number 4. 1 and 4. Name this disaccharide? Well, we kind of started the presentation with that. It's lactose. Number 6, what is the name of the enzyme that cleaves lactose? This is a fun one because it's a little bit of a, it's almost like a typo. Lactase. And this is something that whenever I've taught classes before, people will ask me for short answer questions, does spelling matter? I'll say it does. And that's a tough thing because in this case, when you change the spelling, oh, well, you're actually just changing the molecule. You're not talking about a sugar, you're talking about an enzyme. Okay. So that gets into like the real important nuts and bolts of a disaccharide. Now, to decipher or to process disaccharides a little bit more, well, here we have our glucose, which I always like to be able to look at this and say, okay, we've got carbon 1, 2, 3, 4, 5, and 6. Down, up, down. Okay, so that's glucose. Right, left, right. Now, the tricky part is which variation is this ringed form? Well, let's go back. If our hydroxide group is pointing down, that's the alpha form. So that means that this is beta glucose. And if you want to take it a step further, well, this is pyranose, a six-membered ring. This is a form of glucose that has a ring that has a total of 6 atoms within that ring. Now, we can have a number of different forms of glucose or of a disaccharide. If we look over here, form B and D, okay? Let's look at these names. We've got glucopyranosyl, glucopyranose, glucopyranosyl, fructopyranoside. Okay, so let's see. This is a little bit hairy because if you look at B, let's focus on B for a second. We've got a beta D glucopyranosyl, and then we have 1-4 beta D glucopyranose. So this is a disaccharide made up of 2 glucoses. Okay. Now, what connects these 2 glucoses is a 1,4 linkage. These are both beta sugars. And we know that because, well, we have that name kind of provided out here, beta D, beta D. Now, if we look at this other structure, D, we've got an alpha D glucopyranosyl 1,2 beta D fructofuranoside. Okay, in this case, what we have is, well, carbon number 1 is our anomeric carbon. When we cyclize, C1 is anomeric carbon when cyclizing an aldose, aldose. Okay, but then C2 is the anomeric carbon when cyclizing a ketose, which fructose is a ketose. Okay, so what this shows is we've got the sugar known as sucrose. It's a disaccharide, and it is glucose at carbon 1 linked to fructose at carbon 2. So this is a disaccharide where 2 anomeric carbons link together. This, by virtue of this being two anomeric carbons linking together, this is considered a non-reducing sugar. Whereas, let's look at our other disaccharide. This is, to dig into that other disaccharide for just a moment, this other disaccharide, we have this information here that this is the beta anomer of cellobiose. Okay, now the difference between these two sugars, I'm talking about sucrose versus cellobiose, is, and this is what you have to do whenever you look at a disaccharide, is you have to ask yourself, where are my anomeric carbons? Well, your anomeric carbons, if you remember, are the carbons that previously were not chiral centers, but upon cyclizing, they become chiral centers. Carbon number one on glucose, which is right here, which is also right here. This is non-chiral, but then it's chiral. So that is your chiral center. Now, can you find another chiral center or another anomeric carbon in cellobiose? So this is one. This is our other. So this is an anomeric carbon. Now, the tricky part about this is this has no glycosidic linkage. So this is considered a reducing sugar because of the fact that this right here, this anomeric carbon has no glycosidic linkage. So when we talk about disaccharides, they're classified as being reducing or non-reducing. If they have a free anomeric carbon, like what I see, what I'm showing you at the top, and what I'm going to underline right here, I gotta, yeah, right here. This is a free anomeric carbon. With this being a free anomeric carbon, this is a reducing end of our sugar. Now down here, this does not have a reducing end because both of our anomeric carbons are involved in forming of this glycosidic linkage. So these anomeric carbons, think about them as they're occupied. They are bound to one. So they can't form any other sort of interactions. All right. Now there are lots of sugars, or there are lots of more complicated sugars that are storage, or they're used for storage. Amylose, amylopectin, glycogen, these are all major storage molecules of a bunch of glucose that's been packed together. Now, when you look at these different molecules, if you look at amylose, amylopectin, and glycogen, they all vary a little bit. And what makes them vary basically is the complexity of any sort of branches that they have. Now, with glycogen, we have linear and linear chain plus branches. Amylose, this is kind of like a pigtail, where it just kind of loops and loops and loops and loops and loops and loops. This is just linear. So there are no branches coming off of amylose. Amylopectin, that's linear plus branches. Now, the branches that both amylopectin and glycogen have, they are alpha 1, 4 linkages. Whereas amylose also has alpha 1, 4 linkages. What make, and I'm going to go ahead and draw a bigger box around all of this. What make amylopectin and glycogen different from amylose is that it has not only the alpha 1, 4 linkages, but also alpha 1, 6 linkages. So amylose only has alpha 1 4. Amylopectin and glycogen have both 1 4 and 1 6 linkages. So these complex networks get more complicated based on the presence of these branches. Now, this question at the bottom here, which packs glucose molecules more efficiently? This is a pretty complicated question. Now, the way that, or one of the things that I want you to observe about these is I think that it's helpful to try to identify where it starts. And, you know, and you look at amylose, here's the start, here's the end. Here's the, if you look at amylopectin, let's say that this right here is our start. Well, this is an end, this is an end, this is an end, and so on. So amylopectin and amylose, they both have one start point. But what makes amylopectin different from amylose is amylopectin has lots of kind of ends. So it has, at these start points, we have a reducing end. Amylose has one reducing and one non. I'm going to label it NR or a non-reducing end. Each end on amylopectin, that's a non-reducing end. That's a non-reducing end. Non-reducing end. Okay, now when we look at glycogen, glycogen is a little bit trickier to find out where it starts. And, you know, I'm going to highlight it right here because glycogen is a molecule that has one start. So it has one reducing it. But then, like amylopectin, it has lots of non-reducing ends. Now, whenever I show you this, one of the things that I want you to think about is despite the fact that it has one reducing end and lots of non-reducing ends, what makes glycogen that much different from amylopectin? Now, when you study these structures for just a little bit, I feel like, or I remember this from whenever I took the SAT and whenever I was preparing for the SAT in high school. You would be given a figure and it would be like, which of these, which end of this triangle is the longest? And there would be a note that said, do not look at the scale of the images to calculate the length of a side or anything, and instead you would be provided with here's a measurement and here's the angle and everything like that. But you were told explicitly, don't look at this figure that I've provided you with and think that it means anything. These figures right here are the opposite of that. When you look at these figures, I want you to study them and I want you to figure out like what makes glycogen different from amylopectin. And I know that was a lengthy explanation, but do that for just a second. What I want you to notice, and what I think that you probably did notice, is that glycogen, let's see where, if this is our start point, we've got 1, 2, 3, 4, and there's a branch. And then 5, 6, oh, and there's a branch. And off of that, this second branch, let's count it up. 1, 2, 3, 4. Here's a branch. Okay, so basically every four glucose little balls, subunits, whatever you want to call them, there's a branch point. Whereas amylopectin, let's look at that. 1, 2, branch. 1, 2, 3, branch. 1, 2, 3, 4, 5, 6. Oh, 6. There's a branch. So then if we go over here, we've got basically with amylopectin, we've got a couple where it's every 3 subunits, then we have a branch, then there's one where it's after 6. Now let's look at this next. Let's go over here. 1, 2, 3. There's a branch. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and there's a branch. Okay, so with amylopectin, we had a couple that were as early as 3 subunits in, and then a couple that were it took 7 subunits, or it took 7, yeah, 14, sorry. Um, Whereas glycogen, it's a little bit more consistent. Now, this, so basically, the difference between amylopectin and glycogen has nothing to do with the bonds, but instead branch frequency or branch frequency, branch occurrence, however you want to split that. Now, which packs glucose molecules more efficiently? I think that's going to end up being glycogen, which arguably is good because I like having glycogen so that I can walk around and do stuff. Now, okay, there we go. Now, plants have a storage molecule or storage system of starch. And starch is a combination of amylose and amylopectin. So we already looked at these and we kind of processed them. Amylose is a linear polymer that has nothing but alpha 1,4 glycosidic linkages and glycosidic bonds. Amylopectin is a branched glucose polymer with mostly alpha 1,4 and oh, we already kind of put this together. It has 1,6 branch points, but they're every 24 to 30 units. So they're not very frequent. Okay. Now this is an alpha 1,4 linkage. And this right here is an alpha 1,6 linkage. Between carbons number 1 and carbon number 6 is where you're going to see that branch point. Now, what starch is going to be, it's going to be these two polymers basically kind of threaded together, and they make a very complex network. Now, another so here is our more detailed look at glycogen. Glycogen is a sugar store in animals. It's highly branched. What do we say about glycogen and amylopectin when we looked at them previously? Well, we said that glycogen branches more frequently, and it does. And, our when we were looking at those pictures, we did see that glycogen did branch more frequently than amylopectin. Um, the actual numbers on their frequency is going to vary a little bit, but glycogen is going to have an alpha 1,6 linkage depicted right here, about every 8 to 12 subunits. Now, that's not to say it will, uh, sorry, that's not to say that it will absolutely be every 8 or every 12. And it varies a little bit, which was kind of what we saw in that previous depiction. Now, when it comes to these molecules existing as linear molecules, starch and glycogen, well, they are going to do what, I mean, they're beholden to the kind of properties and principles of intermolecular forces just as anything else is. And so it's nice to say that this molecule is linear, which when we go back a second and look at amylose right here, I said that this was linear. And um, it doesn't take too much of an imagination to look at this molecule and say, that loop-de-loo, and as you described it, um, you, this is from your perspective, so the you is me, um, that curly Q pigtail type of thing, that's hardly a line. It's a very curvy line. This right here is a pretty good model for that. Why would that happen? Well, let's look at this. Let's take a look at the apex of our kind of curl here. What do we have? We got a glycosidic linkage. We've got hydroxyl group, another hydroxyl group. What do we know about hydroxyl groups? O, H. That hydroxyl group's going to have lone pair of electrons. What do we know about this guy right here? That's going to have a lone pair of electrons. This right here, we've also got a hydrogen. So we can have hydrogen bonding that takes place to give us these kind of spiral structures. So when you think about drawing glycogen or amylopectin, it's really awesome and fun to draw it like this, where it's like, aha, here's my straight line. And then here is my branch point and the straight line that follows that. But it's not particularly realistic because what you're doing is you're drawing a nice model, but does glucose look like a perfectly rigid, what would that be, six sides hexagon like that? No, it doesn't. Chances are it's going to take on the chair or the boat conformation. So just keep that in mind that yes, we are going to have hydrogen bonding that takes place whenever we have a so-called linear form of our molecule with only alpha 1,4 linkages. Now, when it comes to roles of these sugars, well, cellulose and chitin are linear polymers of glucose. Or sorry, linear polymers of glucose can exist in the form of cellulose as one, and chitin as another. Now, the biggest difference between cellulose and glycogen and amylopectin is this right here. Rather than alpha, we have beta configurations. Now, that's absolutely necessary to recognize because what do we know about enzymes? Enzymes are extremely specific. Enzymes are selective. If an enzyme says, well, I cut alpha 1,4 linkages and you provide it with a beta structure or a beta sugar, well, that enzyme is going to say, oh, I said that I cut alpha 1,4 bonds. I don't cut beta 1,4 bonds. And so that's important to recognize because, you know, if you if within your gut you had an enzyme that could break beta 1,4 linkages, then you could go outside and break some bark off a tree or pick up a piece of grass, eat it, and you get a bunch of glucose from that. But you don't. You don't have that enzyme. So the enzymes that your body uses selectively will break beta 1,4 linkages, or sorry, alpha 1,4 linkages. They won't touch beta 1,4 linkages. Now, that's one thing that's important to know about cellulose. Another thing is that these cellulose fibers, they consist of around 40 extended glycan or sugar chains held tightly together by an extensive network of hydrogen bonds. Okay, now this figure over here is a little bit complicated. We've got one chain of glucose. We've got another chain of glucose right here, or glucoses, another chain of glucoses. Now I want to erase this because well, I erase all my magical markings there. And what I'd like to highlight are the hydrogen bonds that hold together our sugars. Okay, so here's a glycosidic linkage. Then here's another glycosidic linkage. Here's another. Okay, so if we look back at this, they were kind of repeating elements. It was a nice V, 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 and only whenever we had a 1,6 linkage did we have something other than that. Well, here we've got V and then upside down V. Or if you wanted to, you could say lowercase u and up and then lowercase letter n. We've got these things that are basically opposite of one another. What that allows for is, now I'm going to switch colors, is when we have those beta structures, that enables that oxygen that's found within our ring to hydrogen bond. Again, oxygen within the ring to hydrogen bond. In addition to that, we can also have a hydrogen bond network between strands. So we've got adjacent strands that hydrogen bond with one another. Now, what that allows for is it's almost like, and I'm going to use paper as the example, which is appropriate because we use cellulose to make paper, but it's like this piece of paper, that's one strand. And that strand, it's got tension in it because it's a bunch of hydrogen bonds holding it together from left to right. But, you know, if I really wanted to, I could kind of, you know, if I held tension, I could chop it in half. Okay. But then we add a second paper to it. So each one of these is held together tightly. And then each one of these, like within here, within the two pieces, or between the two pieces of paper, we have a hydrogen bond network holding those two together. So now, if someone is holding tension on this, and if I wanted to chop it in half, is it going to be more easy or more difficult with that second piece of paper? It's going to be more difficult with that, with the addition of that second piece of paper, because now all of a sudden I've got a I, thinking about it like X. So I've got a lateral hydrogen bond network, but then I also have a top-down hydrogen bond network. And these are just sheets and sheets. And that's what's going to give strength to things like trees and any sort of plant stock or anything like that. Okay, so that's cellulose. Beta 1,4 linkages. We don't have the enzymes to break those beta 1,4 linkages. And in addition to that, the orientation of those beta linkages allows for adjacent sheets to basically stack on top of one another. So the complex part about this is even if we did have the enzymes that were capable of breaking beta 1,4 linkages, we would still have to combat and basically offset the uh, the hydrogen bond network that is otherwise there. And so then it's like, well, in an aqueous environment, that hydrogen bond network is still going to be pretty strong. Um, so that's what the important parts about cellulose are. Now, chitin is a similar polymer made up of, rather than glucose, it's N-acetylglucosamine. Now, chitin is the predominant polysaccharide or polymer of like exoskeletons for beetles and stuff. Okay, glycoconjugates. There are three main classes of glycoconjugates. There are proteoglycans. I like these just because the words sound fantastic, or they are fantastic. Peptidoglycans and glycoproteins. I just feel like I'm in a sci-fi movie, like I'm saying, which I'm not. I'm in a non-fi movie. Um, the protein is attached to a particular type of polysaccharide called a glyco or glycosyl like glycosaminoglycan. Though, or by weight, proteoglycans are mainly carbohydrates. Peptidoglycans are polysaccharides linked to small peptides. So this is a part that is, well, peptides and sugars. Glycoproteins are kind of like peptidoglycans, but they're much bigger. The proteins that have covalently bound oligosaccharides, a protein is the largest component by weight. So that's where we have a little bit of a difference there. So, yeah, glycoconjugates. It's a hybrid of a sugar and a protein. But the question is, where is the weight mainly distributed? Proteoglycans, it's your sugar, is where the weight is distributed. Glycoproteins, the protein is the biggest component of it. Now, glycosaminoglycans and proteins. There's a couple of these. Heparin hyaluronate, dermatan sulfate, chondroitin 6 sulfate, keratin sulfate. These proteoglycans are major structural components and are very strongly and tightly held together. Peptidoglycans. So many cell walls contain peptidoglycan. The cell wall on the outside of gram-positive bacteria uh, or is going to have peptidoglycan. Crystal violet is the stain that can be used to detect, and if you've taken like a microbiology class, then you probably did a crystal violet stain, which helps you identify whether or not that peptidoglycan is there. Um, and basically during the synthesis, 5 amino acids are added to the sugar, and essentially you end up with sorry, this is what I meant. Um, these You have this network of this this sugar network as well as this peptide. So here's your sugars. So it's going to be repeating elements of this over and over and over again. Then this backbone right here, which consists of L-alanine, D-isoglutamate, L-lysine, and D-alanine. So the interesting thing about that is whenever we think about amino acids as your naturally occurring and um, when you think about the naturally occurring forms of an amino acid, typically those are your L amino acids. Um, D amino acids are typically not your proteinogenic amino acids, but we have this mixture here. Then there's this 5-glycine or a pentaglycine bridge that links to the next peptide. So peptide right here linked to, well, a pentaglycine links to another peptide, or the other peptide repeated over and over again with these sugars basically making up that that peptidoglycan layer. Now, glycoproteins are secreted or they're membrane proteins, and these have some pretty cool and complex structures. So these are secreted and or they're membrane proteins. They have N or O linked to amino acids, or they are N linked or O linked to an amino acid. Now, what that means is that we have some modification to an amino acid. So this blue blob right here, this is your protein. Now, that protein is going to have either an asparagine or a serine. Now, that asparagine is going to have a N linkage. So what that means is that on your asparagine side chain, which, okay, so here is our alpha carbon. Here is our R group for asparagine, which is CH2, C double bond O, and then NH. Okay, so there's my amino acid side chain. To that, we're going to add to it a modified form of glucose. That GLC indicates glucose. You'll notice GLC and GAL are different sugars. GLC is glucose, GAL is galactose. Now, this is my anomeric carbon linked to the nitrogen within my R group. Okay, so here is glucose, and to that glucose, we have an N-acetyl group, hence the NAC. So then we could also have a GalNAc, which is galactose plus an N-acetyl group. Now, the difference between these is that, well, serine is the place where this is going to be anchored. Now, these GLICNAc or GalNAc, they are going to be N-linked or O-linked. GLICNAc has to be N-linked and GalNAc has to be O-linked. The amino acids upon which they are added to are going to vary a little. Now, the complexity of these linkages is going to vary a little bit. Asparagine will always be the site for the GLICNAc. Serine will always be the site for the GalNAc. Now, what we have over here, this figure right here, shows that as soon as that GalNAc, or that GLICNAc, because notice this is asparagine, 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 receives the GLICNAc, then what we can also have added to it is another GLICNAc. So basically what there's going to be is another, I'm going to do some erasing. Another GLICNAc can be added right here to carbon number 4. Well, that is going to be an O-glycosidic linkage where we're adding another one of these whole things, but it's going to be added right here. What I want you to take away from this is that this is basically a network that we can make of sugars off of a single asparagine or a serine. What I want you to know are what are these sugars that are going to be added to either one of these amino acids, and what are the linkages? They're either O or N glycosidic linkages, depending on the R group. Serine has an oxygen, it has a hydroxyl group. Asparagine, well, that has the uh, carbonyl carbon and an NH group, so that can be the source of our um, N linkage. Now, these make almost pitchfork looking sugar structures. Those sugars way out there, what they can be used for is basically communication. They can be kind of projected from a cell to say, hey, I'm this type of cell. And so then other cells nearby say, oh, well, I see there's a bunch of glycnacs on there. Or I see this out here, for example, there's a salicylic acid out there. Okay, so salicylic acid is projected from this cell, so that tells me this, that gives me some information. Because remember, cells largely are going to be kind of simple machines. If they see one thing, then they're going to say, oh, well, that's what that is. Moving on. So these glycoproteins communicate. And sometimes it's a message of like, oh, we're a stress cell, so we need to undergo or initiate senescence. Or we're a developing cell, so we're going to have a bunch of these glycoproteins displayed on the surface. Now, oligosaccharides, in addition to serving as storage or structure, um, this is kind of a continuation of what we were just talking about here with glycoproteins, but oligosaccharides themselves can be used for recognition. So complex sugar structures at cell surfaces are recognized by specific proteins known as lectins. These proteins tightly bind specific structural features by extensive hydrogen bonding. So whenever you see this, I want you to think about, well, what are the amino acids that would most likely participate in this hydrogen bonding? Histidine, asparagine. I'm going to switch from that. Histidine, asparagine, and arginine, glutamic acid, arginine. Those are all strong candidates for hydrogen bonding. Now, there are different structures, different sugar structures found in glycolipids, and these are also produced and displayed on the surface of a cell. And these are going to be what are helpful for discerning different blood types from one another. So depending on basically the network or the complexity of the sugar, that's going to designate a sugar as an O, A, or B antigen. So the B antigen, for instance, is going to be glycnac, uh, galactose, and then a fucose. Whereas the A antigen is glycnac, galactose, with a fucose coming off of this, and then a galnac. So the sugars are different from one another. Okay, what I want to there's lots of things that I want you to be familiar with here with this chapter. I would definitely know the different roles that sugars can play. Definitely know the types of interactions and bonds that they can have. My background is in plant biochemistry, so I kind of tend to be curious about uh, cellulose, make sure that people know the like differences between cellulose and galactose, or sorry, cellulose and glycogen, because those are important things. It's not just beta linkages, it's beta linkages and what do those beta linkages kind of accommodate or propagate. All right, I hope this is helpful, and I hope you have a good one.